Hello, everyone, and thank you for listening. Today, I would like to introduce stimulus-responsive copolymers designed for precision medicine. I will discuss how these smart materials are reshaping drug delivery, enabling treatments that are more targeted and less toxic, while achieving unprecedented levels of precision. Let's start with the fundamental question. What exactly are environmental responsive copolymers? Simply put, they are a class of smart polymeric materials that can sense and respond to specific biological or environmental stimuli. When they encounter a trigger, such as a change in pH, temperature, or the presence of certain enzymes, they undergo physicochemical changes. These changes can include conformational transitions, solubility shifts, bond cleavage, or swelling and dwelling. Why does this matter for drug delivery? Because these changes can be harnessed to trigger controlled drug release precisely at the target site, whether that a tumor, an area of inflammation, or a specific intracellular compartment. The key advantages are clear. Targeted release at disease sites, reduced systemic toxicity and side effects, improved bioavailability, and spatiotemporal control over drug release kinetics. In essence, these copolymers act as molecular switches, smart enough to know when and where to release their therapeutic cargo. So, how does this actually work? The process unfolds in four sequential steps. First, stimulus recognition. The copolymer senses environmental changes, such as pH shift, temperature change, redox potential, enzyme activity, or light irradiation. Second, the polymer chains undergo a conformational change. They may swell, collapse, cleave specific bonds, or switch their hydrophobicity. Third, this structural transition causes nanocarriers, micelles, nanoparticles, or hydrogels, to destabilize and disassemble, exposing or releasing the encapsulated drug. And finally, the therapeutic payload is liberated at the target site with tunable kinetics and spatial precision. It's an elegant chain of events. Detect, transform, disassemble, release. Now, let's look at how these systems are classified. We can categorize them based on the type of stimulus they respond to. pH-responsive copolymers rely on protonation or deprotonation of ionizable groups and acid-label bonds. Examples include PA and PDMEMA. Temperature-responsive systems exploit LCSD or UCSD phase transitions, like NIPAM and PGPCL. Redox-responsive copolymers use disulfide bond cleavage under reducing environments rich in glutathione. Light-responsive systems use photoisomerization or photocleavage triggered by UV, visible, or near-infrared light. Enzyme-responsive copolymers incorporate peptide linkers cleaved by specific enzymes. And finally, multi-stimuli systems combine two or more triggers for enhanced specificity. Each category has its own strengths, and the choice depends on the target disease microenvironment. Let me now walk through each major stimulus type in a bit more detail. pH-responsive copolymers. These exploit the pH gradients that exist between healthy and diseased tissues. Normal blood has a pH of 7.4, but tumor microenvironments are more acidic, around 6.5 to 6.8. Endosomes and lysosomes are even more acidic, at pH 5.0 to 5.5. By incorporating protonatable groups or acid-label bonds, such as hydrazone or acetal linkages, these copolymers remain stable in blood but disassemble in acidic environments. A classic example is PGBP-DMA, micelles, which stay intact at pH 7.4 but release doxorubicin when the pH drops below 6.5, selectively killing tumor cells. Temperature-responsive copolymers. These operate through phase transitions at a specific temperature known as the LCSD. Below the LCSD, the polymer is hydrophilic and swollen, holding the drug in. Above the LCSD, it becomes hydrophobic and collapses, expelling the drug. The gold standard here is NIPAM, with an LCSD of about 32 degrees Celsius, very close to body temperature. By tuning the copolymer composition, we can shift the LCSD to exactly 37 degrees, enabling body temperature trigger release. Redox-responsive copolymers. This approach exploits the dramatic difference in glutathione concentration between the extracellular and intracellular environments. Outside the cell, GSH is only 2 to 20 micromolar, so disulfide bonds stay intact, and the drug remains encapsulated during circulation. But inside the cell, especially in tumor cells. GSH concentrations are 1 to 10 millimolar. That's 100 to 1,000 times higher. This causes rapid disulfide bond cleavage and intracellular drug release. It's a remarkably effective selectivity window. Light-responsive copolymers. Light offers something unique: remote, non-invasive activation with precise spatial and temporal control. There are three main mechanisms: photoisomerization, using azobenzene, photocleavage using coumarin or O-nitrobenzyl groups, and photothermal conversion, using upconversion nanoparticles. Near-infrared light is particularly attractive because it penetrates deeper into tissue with less phototoxicity and enables on-demand, pulsatile drug release simply by switching the light on and off. Enzyme-responsive copolymers. These systems take advantage of enzyme overexpression in disease pathology. For example, matrix metalloproteinases are overexpressed in tumors, cathepsins are elevated in lysosomes in cancer cells, and beta-galactosidase serves as a bacterial marker in the gut. By designing short peptide sequences as cleavable cross-linkers, we achieve high selectivity. The copolymer only disassembles when a specific disease-associated enzyme is present. Now, here's where it gets really interesting. Single stimulus systems can sometimes suffer from premature release or insufficient specificity. Multi-stimuli responsive copolymers solve this by implementing ANOR logic gates. The drug is released only when the correct combination of triggers coexists. For instance, a pH plus redox dual system releases its cargo only when it encounters both tumor acidity and intracellular GSH. A pH plus temperature system enables both passive targeting and externally activated release. And a triple responsive pH redox light system offers maximum control, spatial, temporal, and biochemical. This multi-gated approach dramatically reduces off-target toxicity because the probability of all triggers being present simultaneously in healthy tissue is extremely low. Let me show you how this plays out in cancer therapy, arguably the most actively pursued application. The tumor microenvironment provides a unique combination of triggers: acidic pH around 6.5, elevated GSH at 1 to 10 millimolar intracellularly, MMP overexpression, and hypoxia. Smart copolymers exploit these features through multiple strategies. pH-responsive PGB-BA cells deliver doxorubicin, releasing upon endosomal acidification after accumulating in tumors via the EPR effect. Disulfide cross-linked nanogels encapsulate paclitaxel and rapidly disassemble in the GSH-rich tumor cytosol. Dual pH redox nanoparticles combine hydrazone and disulfide linkers for cascade release. And by functionalizing these copolymers with targeting ligands like folate or RGD peptides, we further enhance tumor uptake. It's a multi-layered targeting strategy, passive, active, and stimuli-responsive all working together. Beyond cancer, these smart materials are making impact across many therapeutic areas. In gene delivery, pH-responsive polycations condense DNA or RNA into polyplexes and use the proton sponge effect for endosomal escape. For anti-inflammatory therapy, enzyme-responsive copolymers release corticosteroids and inflamed tissue. In wound healing, temperature-responsive hydrogels form in situ gels at body temperature for sustained release of growth factors. They're also being used for oral drug delivery, protecting acid-labeled drugs like insulin in the stomach and releasing them in the intestine, as well as in tissue engineering scaffolds and antimicrobial delivery systems that respond to infection-specific microenvironments. On the technical side, these systems are built using controlled polymerization techniques like RAFT, ATRP, and ROP, which give us precise control over molecular weight and block sequence. Amphiphilic block copolymers then self-assemble into micelles, vesicles, or nanogels in solution. Click chemistry allows modular attachment of responsive linkers and targeting ligands. And scalable techniques like nanoprecipitation produce uniform particles in the 50 to 200 nanometer range, ideal for biomedical applications. Of course, this field is not without challenges. The advantages are compelling: site-specific release, reduced toxicity, enhanced efficacy, and on-demand control. But we must also address real hurdles: biocompatibility and long-term safety of synthetic polymers, batch-to-batch reproducibility, GMP manufacturing scalability, premature drug leakage during circulation, limited tissue penetration for light-triggered systems, and the gap between in vitro and in vivo behavior. These are active areas of research, and progress is being made on all fronts. To summarize, environmental responsive copolymers represent a paradigm shift in drug delivery, moving from passive carriers to intelligent systems that can sense, decide, and act. By harnessing the unique microenvironmental signatures of disease, these smart polymers are bringing us closer to the ultimate goal of precision medicine: the right drug in the right place at the right time at the right dose. Thank you very much for your attention.